Polypeptide synthesis is the production of peptides. Polypeptides are chains of amino acids joined together by peptide bonds. The correct sequencing of these amino acids can create proteins. The central dogma states that the process of gene expression occurs in one direction and in two steps: transcription and translation. Phase 1 is initiation. The DNA itself is too large. It must create a smaller version of itself. When a gene is switched on, the enzyme RNA polymerase binds to the promoter of the DNA, which is the start of the region that's going to be copied. Enzyme DNA helicase comes and unzips the double-stranded helix, breaking the hydrogen bonds between the complementary bases. In reality, smaller regions of the DNA are exposed. Phase 2 is elongation. This is when the RNA polymerase moves along the antisense strand in the 5' to 3' direction, reading the bases and adding complementary nucleotide bases to the RNA, whose chain grows in the direction of also 5' to 3'. This is to replicate the sense strand. There is a difference in nucleotide base pairings. Thymine in DNA is replaced with uracil in RNA. The RNA continues elongating. Phase 3 is termination. At the end of a gene, a sequencing of nucleotides called the terminator is located. When the RNA polymerase reaches the terminator, a termination signal stops it from transcribing any further and tells the RNA polymerase to disconnect. The DNA strands are closed up again by enzyme ligase. Now, the premature messenger RNA or mRNA must undergo co-transcriptional processing or editing to become a mature mRNA. There are 3 main editing processes. Splicing is when the non-coding regions of the gene, called introns, are removed with the help of RNA protein complex snRNPS. These spliceozymes bind to the introns, make them fold and remove them. Then the exons, which are the coding regions, join together. 5' capping is a process when a 7-methyl-guanosine, a modified guanine nucleoid, caps the 5' end of polyadenylation is when an enzyme called polyadenylate polymerase catalyzes a string of adenine nucleotide bases, attaching them to the 3' end of the pre-mRNA. This is called a poly-A tail. Fun fact, a poly-A tail can be 250 nucleotides long in eukaryotic cells. The mRNA must mature because cytoplasmic enzymes, ribonucleases, and other exonucleases can damage the mRNA's ends by cleaving down the phosphorus-oxygen bonds. Hence, splicing, the G nucleotide capping, and poly-A tail protect the mature mRNA. Termination in transcription is now complete. The mRNA then travels out of the nucleus through a nuclear pore, wanting to intersect with a free ribosome or one attached to the ER in the cytoplasm. At the same time, transfer RNAs are being activated in the cytoplasm. The enzyme amino-acid tRNA synthetase acts like a tRNA ligase and together with ATP catalyzes the amino acid with its complementary uncharged tRNA molecule, creating an amino-acid tRNA. The tRNA then becomes charged and can be used in the translational process. Now, translation's first phase, initiation, begins when a ribosome recognizes the mRNA's start codon. What is a codon? A codon is a sequence of 3 nucleotide bases that code for a specific amino acid. As seen here, the ribosome's small and large subunit wraps around this mRNA start codon, AUG, which is the same for all mRNA strands. Ribosomal RNA will assist in the subunit process to create the translation complex. tRNA molecules themselves have an anticodon region that's complementary to the mRNA's codons. So once the codons are read by the ribosome, the tRNA with the complementary anticodon comes and joins with the mRNA, carrying the specific amino acid on top. Initiation is completed once the tRNA carries the amino acid for the start codon, AUG, which is methionine. Elongation starts when the ribosome moves along the mRNA to the second codon, and another tRNA with the complementary anticodon connects with the mRNA's codon. The amino acids at the top will start joining together. Hence, the methionine will join with another amino acid, forming a peptide. The empty tRNA will then leave the ribosome. The ribosome now is split up into 3 sites. Site A, where the tRNA enters the ribosome, site P, where the peptide bonds are created, and site E, where the empty tRNA leaves the ribosome. Elongation continues until the stop codon, in this case UAG, is reached. Termination occurs as the UAG stop codon is detected by the class 1 release factor protein, which then binds to site A, signaling for the release of the new polypeptide chain. When the polypeptide is released, the translation complex disassembles, splits into its subunits, and the ribosome can now bind to new mRNA. The released polypeptide chain undergoes structural change from the primary to secondary to tertiary to quaternary structures to form a complex 3D protein that's functionable. The mRNA strand then breaks down and the free nucleotide bases can be reused. To summarize, transcription is the first stage of gene expression where genetic information is copied from DNA's double helix to a single-stranded mRNA, which can leave the nucleus. Transcription occurs in 3 stages: initiation, elongation, and termination. Translation is the second process of gene expression. It is when the mRNA is read by a ribosome and, with the help of tRNA, is transcribed into protein. Thank you very much.